So this discussion is going to be about this paper, the association of spinal fluid glucocerebrosidase activity with the risk of incident dementia in patients with Parkinson's disease by Dr. Ofdal et al. From the Park West study in Norway that's been going on for several years.
Hi, everyone. My name is Avner. I am a neurologist and a researcher, specifically in the genetics of Parkinson's disease in the movement disorder unit at the Tel-Aviv Medical Center in Israel.
And I am Joseph Friedman. I am a clinical neurologist and a clinical researcher who's had a longstanding interest in behavioral aspects of Parkinson's disease and have been involved in many clinical trials for the treatment of various aspects of Parkinson's disease. The first question is, why was this study done? And the reason is that GBA mutations turn out to have a very important role in our understanding of Parkinson's disease. That role hasn't been fully explicated, and this is one attempt to do that. About 30 years ago or so, it was recognized that people with Gaucher's disease have an increased incidence of Parkinson's disease, and a number of mutations have been recognized which are linked to Parkinson's disease. In fact, there are over 300 presumed pathogenic genes that have been implicated. They have variable penetrance, some being associated with a higher risk of developing Parkinson's, some not. The Ashkenazi Jewish group has the highest incidence, I think, that's been identified so far, with an abnormal gene present in between 10 and 31% of the population, which is several times more than other populations it's been compared to. But nevertheless, the pathological gene only increases the risk of Parkinson's disease to somewhere between 8 and 15%. And this raises some interesting questions because the gene is just a single gene, whereas to have Gaucher's disease, you have to have to be homozygous for this. And yet the risk in Gaucher's disease is about the same as the risk for having an isolated mutation, pathogenic mutation. There are differences between GBA-associated Parkinson's and non-GBA-associated Parkinson's in general, with an earlier onset and an increased risk of hyposmia, REM sleep behavior disorder, and dementia, which is what precipitated this project. Dr. Thaler, can you discuss the sort of the genetics and the what's understood about the pathophysiology of this disorder and then the study itself?
Definitely. So the GCAS is an enzyme that is part of the sphingolipid metabolism pathway, which we can see on the screen here. This pathway has several mutations that are associated with it that cause different diseases. And as you said, Gaucher disease is caused by a mutation, a homozygote mutation in the GCAS gene. And indeed, from Gaucher's disease and first-degree relatives of patients with Gaucher, it has been established that heterozygote carriers of GBA mutations do have an increased risk for Parkinson's disease and a worse disease phenotype compared to patients that do not harbor mutations in the GBA gene. And there has been a bidirectional loop associating mutations in GBA and alpha-synuclein accumulation. Alpha-synuclein is the pathological hallmark of Parkinson's disease, and reduction in GCase levels increase the aggregation of alpha-synuclein, which in turn continues to reduce GCase level, which is a certain vicious circle. The study that we are discussing, which came from Norway, it's part of the Park West study, aimed to assess GCAS activity within the CSF, within the cerebrospinal fluid, taken from 117 PD patients and 50 age-matched controls. It was a 10-year follow-up study which assessed individuals with Parkinson's disease between each year and every other year, and the controls were assessed only at baseline. Parkinson's disease dementia was clinically diagnosed, and initial GCase activity within the CSF was analyzed. All PD patients were assessed for mutations in the GBA gene. And we can see the characteristics of the cohort in the table that is presented. Joe, would you like to discuss the results?
Well, the results are very, very interesting because there was an association made between a reduced amount of GBA activity, indicating pathological GCase activity, with the development of dementia. But yet there was no significant difference between the GCase activity between healthy controls and people with Parkinson's disease who did not have dementia. The association of the GCase activity had an inverse relationship with the development of dementia. So lower activity, meaning more impaired enzymatic activity, was associated with an increased risk of dementia. And that is the major finding here, but there were certain limitations here, including, as you could see by from the box plots, that there's a significant overlap in the activities measured between any two groups, even though there were statistically significant differences between healthy controls and most of the groups. So that was a very interesting findings. But there are certain, and this is what the researchers were in fact trying to show, and this was their hypothesis. But there are certain limitations to this study, including the small numbers. So here's a Kaplan-Meier survival graph showing that just looking at GCase activity, the fall off was greatest. That is, the most patients became demented, or their risk of dementia was highest if they were in the lowest third of GCase activity compared with the other groups. But the numbers were relatively small, and there was a high dropout rate because a fair number of these patients actually died. So there were only 12 patients in this study who were GBA positive, which is not all that surprising since the population in Norway does not include many Ashkenazi Jews. The GBA status wasn't assessed for controls, and we don't know what happened to the controls because they were only seen once. And among the patients who became demented with Parkinson's disease, we don't know how many of those had GBA mutations versus the others. So whether the 12 were mostly GBA or not is not stated in the report. And another interesting thing, which might likely be actually in another paper, which might be in press for all I know, from the Park West, is whether or not the GCase activity had any relationship to the progression of motor symptoms. So if we assume that GCase activity is related to alpha-synuclein accumulation, that is, aberrant alpha-synuclein is not cleared because of aberrant GCase activity. And so Lewy bodies, or at least abnormal alpha-synuclein, accumulates, we would expect to see worsened motor function as well, and we don't have that data. I should point out that the Park West group has published a large number of papers. I just did a quick review and found 36 papers that they've published since they started the study, most of which are related to behavior like dementia, but not all. And I would not be at all surprised if they have other papers in the works answering this question.
Yes, the methodology of GCase assessment in CSF is very controversial. There have only been very few papers that managed to assess the levels of the enzyme activity within CSF. In this paper, the enzyme level was corrected for total protein, but the origin of GCase within CSF is not totally clear. One possibility is it comes from membranes of cells that are found within the CSF, and that was not mentioned in the paper, and it wasn't corrected for. In different papers, the levels of GCase activity within CSF varied very much. There is one Italian group that has done a lot of research within GCase activity in CSF, and their reported levels were universally lower than the levels in this paper. Here we actually see within Ashkenazi Jews the breakdown of the genetics that are associated with Parkinson's disease. This is from the center I work in. We followed up the first 1,200 PD patients from total Ashkenazi background, and we see that there is about 18% that harbor mutations within GBA, and these are further separated into mild and severe mutations. But again, in Norway, we don't have the typical Ashkenazi mutations, and the frequency of the mutations should be much lower. And again, another limitation is that we don't know the genetic status of the 50 controls they used. Within these limitations, there are a lot of interesting questions that the publication brings up. First, the GBA status didn't seem to be relevant to the findings. It was the GCase activity disregarding GBA status. We do know that GBA PD and idiopathic PD or non-GBA PD seem to have relatively similar symptoms. They're both DOPA responsive. Again, GBA PD tend to have worse phenotype and a more faster deterioration, both motor and cognitive wise. There is a large PPMI paper coming out depicting synuclein seeding assays that were done for the whole PPMI cohort, in which GBA PD and idiopathic PD have a very high rate of positive seeding assays as opposed to LARC2 PD. And this being a good target for drug development and personalized medicine, there has already been at least one study by Sanofi that targeted GBA, even though it failed. And we do have several studies that are currently underway or are supposed to start in the near future. Current treatments for Gaucher's disease have not been shown to be relevant for Parkinson's disease, mainly because they currently do not cross the blood-brain barrier. And we do know that from post-mortem studies that GBA activity within brains of idiopathic Parkinson's disease are also lower. GCase activity is also lower compared to patients or compared to controls without Parkinson's disease. Joe, I will let you.
Yeah, it raises interesting questions. So there is a study planned or possibly going on right now of this medication, Ambroxol, which is a mucolytic agent, which is used for treating people with upper respiratory infections to help them clear their phlegm, which is thought to act by altering chaperone proteins for GCase activity to make it to increase the activity. So it will be a treatment targeted at slowing progression of dementia or prolonging non-demented state in people with Parkinson's disease. But GCase activity has also been implicated in some studies looking at dementia in frontotemporal dementia and also in the dementia associated with ALS, in which case the effect on alpha-synuclein may not be all that important, and drugs like Ambroxol may be helpful for other dementias as well. Although this has potential in terms of predicting which patients are most likely to develop dementia early on in Parkinson's disease, I think the problem that the box plot graph illustrates is the large overlap between Parkinson's disease without dementia and Parkinson's disease with dementia. That although there's a statistical difference between the two populations, the overlap is large enough that with any particular patient who you choose, you're not going to have a high degree of confidence about which group they're going to fall into. Certainly, using GCase activity will allow you to increase the likelihood, but how much it will be increased really is hard to determine, and partly that's due to the small population that they have. So that's the question that I have. There are, a number of things that have been identified that increase the risk of dementia in people with Parkinson's disease, and whether this will turn out to be more important than these other predictors remains to be seen.
And again, if we had data regarding the peripheral GCase activity that would, whether it correlated or it did not with the CSF GCase activity, that would be interesting, as ascertaining peripheral GCase is much easier than lumbar puncturing our patients.
Sure. But nevertheless, it represents a very interesting step forward, I think, and hopefully this can be reproduced in larger populations, although, as you point out, doing large studies of a large number of patients with spinal fluid is challenging. This
was our assessment of the association of CSF Glucocerebrosidase activity with risk of incident dementia in patients with Parkinson's disease.